Hi, it's Mrs. Vance again, and we're on to Part 4 of Unit 12 Notes, which is about bacterial reproduction and nutrition. So, there are several different ways that bacteria can reproduce. The main way bacteria reproduces by binary fission. This is asexual cell division. This is the kind that can happen as frequently as every 20 minutes, usually not much more than an hour. It doesn't take very long to go from just a few bacteria to millions of them. There are several kinds of, we can call it sexual reproduction. It's not really sexual like we think about sexual, but basically reproduction that involves recombination of genes from different sources. And so we'll talk about each one of these. Transformation is when bacteria pick up DNA from another source. Conjugation is when they exchange genetic information, usually in the form of exchanging a plasmid. Transduction is when genes get accidentally transported from one bacterium to another via a bacteriophage virus. And endospores, we'll consider that in here as well. These are those resistant cells that can allow the bacteria to withstand harsh conditions. And so let's talk about these. So binary fission in bacteria is really very, very simple, much more simple than mitosis. Basically, the cell is getting bigger, and at some point the DNA gets replicated. Each piece of DNA, notice that it here attaches to the cell membrane, and as the cell continues to grow, basically the two copies of the genome get separated from each other. The plasma membrane starts to divide along with the cell wall there, and then a cross wall forms between the two cells, and then you have separate cells there. And so it's really, really very, very simple. This is actually a picture of a Bacillus licheniformis starting to divide. Okay, and you can see the partially formed wall there and the two separate genomes. In terms of genetic variation, so transformation is when the bacteria picks up DNA from its environment. It can be DNA that's been left behind by dead bacteria. This is the process we use for genetic engineering, when we actually have a particular plasmid with genes on it that we want the bacteria to make or to do something with, and we put them in a situation that makes it easy for them to pick up the DNA. Bacterial transduction happens by means of a phage. Bacteriophage DNA, sometimes when it releases from the lysogenic phase and goes back into the lytic phase, can actually sometimes cut off pieces of the bacterial DNA as well. And so when the phages are released, they might carry some additional genes from one bacterium to another, if the bacteria happens to survive it. Bacterial conjugation is when you have a plasmid that basically gets transferred purposely from one bacterium to another. There's a special kind of pilus that can be produced called a sex pilus that's basically like a hollow tube, and that allows the donor cell to transfer a plasmid from it to the recipient cell. And that's one way that a lot of bacteria pick up antibiotic resistance, which is becoming more common in a lot of bacteria. Prokaryotes have a lot of different varieties of ways that they can feed themselves. Okay, and so we need to talk about some more complex things here. In animals, we generally talk about, or animals and plants, we generally talk about, autotrophs and heterotrophs. But we have to define that a little bit more precisely when it comes to bacteria. So first of all, we'll talk about how their energy source. So phototrophs capture energy from sunlight. The photo means light. And chemotrophs harness their energy that's stored in chemicals. We would be chemotrophs, for instance, because we use energy that's stored in chemical compounds that we ingest. There are also different sources of carbon that can be used. And so autotrophs get carbon from carbon dioxide. Heterotrophs get carbon atoms that are present in organic compounds from other organisms. So we would be heterotrophs in this case. We would be chemotrophs and heterotrophs. So you can combine those together, okay, that describe how they get their energy and their carbon. So photoautotrophs get energy from sunlight and use carbon dioxide for carbon, like plants do. Now we're talking about prokaryotes here, so this would be something like cyanobacteria that are other photosynthetic bacteria. Photoheterotrophs get their energy from sunlight, but their carbon atoms from organic molecules. Okay. Chemoautotrophs harvest energy from inorganic chemicals but use carbon dioxide for their source of carbon. And chemoheterotrophs get their energy and carbon from organic molecules. So plants would be photoautotrophs, although they're not bacteria. Animals would be chemoheterotrophs because we get energy and carbon from organic molecules. But again, we're not bacteria. These categories really only apply to bacteria. So you could make a little chart basically that lets you compare photoautotrophs to chemoautotrophs and so forth as far as their carbon source and their energy source. It would look something like this as a way to compare them. And some examples of these bacteria are like this. So there's one called Oscillatoria that's actually a photosynthetic bacterium that lives in strands like this. This is some kind of rock-eating bacteria, basically, that's a chemoautotroph. Rhodopseudomonas is one that is a photoheterotroph. It uses energy from sunlight, but it's carbon from organic molecules. And then chemoheterotrophs, here's one called Delavibrio that's attacking a larger cell here. But those get their energy and their carbon from organic molecules. Bacteria can also have different metabolic relationships with oxygen based on whether they can use oxygen or not. And so learning these terms is really important here. Obligate aerobes require free oxygen. The word obligate, think about obligated. When you're obligated to do something, you're required to do it. Okay, so they require free oxygen, like we do. We are obligate aerobes. Facultative anaerobes use oxygen if it's present, but if not, they can use fermentation. Facultative means it comes from the same root word that facilitated comes from. Basically, they can be either anaerobic or aerobic. They can do whatever works for them at the time, whatever is present at the time. And then obligate anaerobes are ones that are poisoned by oxygen, and their only source of energy would be from fermentation or from anaerobic respiration of some kind. So obligate means it's required, and so you've got obligate aerobes and obligate anaerobes, and then you've got facultative anaerobes that can use oxygen or not, depending on what's present. And that's ending the notes on nutrition and reproduction.